This video is on implant-associated infections. The learning objectives are to understand the source of microorganisms that cause implant-associated infections, to know some of the microorganisms that are most commonly responsible for implant-associated infections, and to understand the steps to infection and the problems that are presented when you get biofilm formation on an implant. In the previous video, we talked about implant-associated infection, and we said that 1 to 6% of implanted devices become infected, and this represents a huge cost to the healthcare system and to patients as well. And the question that we posed at the end of that was that how is this possible when our devices have to be sterilized to achieve a sterility assurance level of 10 to the minus 6, meaning that only 1 in a million of these devices after sterilization treatment would contain any microorganisms that could cause an infection. The reason that it happens is because there is transformation of bacteria that are usually found on or in the body. So in a surgical procedure, of course, the physician is going to sterilize the area before making any sort of incision. But we can't completely eliminate all of the microorganisms from the human body. So these ones, we naturally have a community of microorganisms known as the microbiome in our body and on our body. And they're normally innocuous, so they're not harmful. But in some cases, when we have an implant, there is a transformation that occurs where they become virulent once they get on the surface of a biomaterial. Now, before we go into how that process happens, I want to talk about some of the most common offenders. And we're going to break them up into gram-positive bacteria, gram-negative bacteria, and fungi. And these are some of the important species here. So for gram-positive bacteria, you have Staphylococcus aureus and Staphylococcus epidermidis. So these are found on skin. They're on our skin normally. And then gram-negative bacteria, so these are Enterobacteriaceae. So this is in the gut or the enteric system. These can be found. And then Pseudomonas aeruginosa, which can be found on our skin as well. And then we'll talk about for these some of the devices that can be affected by them as well. And then Candida is also found on our skin and mucosal surfaces. But first, a little bit of background on gram-positive and gram-negative bacteria. So this terminology just has to do with the structure of the cell wall, but specifically it's derived from the stain that is used to visualize bacteria. So in the field of microbiology, there is a technique known as gram staining that's used to visualize bacterial cultures, kind of analogous to what we do in histology when we stain our mammalian cells. But this gram stain has a characteristic appearance depending on the cell wall structure of bacteria. So gram-positive bacteria have a cell wall that has many layers of this peptidoglycan. So this, it's very thick, as is shown up here. And then, but that gram stain is able to penetrate through that, and then it stains the cells kind of a purple color, I believe. In contrast, for gram-negative, the cell wall is thin, but it's kind of this multi-layered structure. So the inside, it's made of the peptidoglycan that's similar, but then it has this outer membrane composed of phospholipids and lipopolysaccharides. So lipopolysaccharides in particular, they stimulate a lot of inflammation. So these gram-negative bacteria, they tend to be more antibiotic resistant. They also tend to be more virulent. Now let's talk about some of our common offenders. So first, we have Staphylococcus aureus, which is one of our gram-positive bacteria. Staphylococcus bacteria are described as having a characteristic appearance that's like clusters of grapes. So you can see that if you look up at the top here. So this is a scanning electron micrograph of Staph aureus that's growing on a surface. And then here, this is just light microscopy down here. And you can see this purple stain, so that's the Gram stain that I mentioned on the previous slide. So the Staphylococcus aureus is an important one because it is the most common bacteria that's associated with orthopedic implant infections, so your knee and hip replacements. As our example of a gram-negative bacteria, we have Enterobacteriaceae. So as I said earlier, this is a family of gastric bacteria or gut bacteria. So it includes Escherichia coli or E. coli and Salmonella species. So here again we have SEM, and then this is our light microscopy picture. So you can see these have more of a rod-like appearance as opposed to the spherical appearance of the Staphylococcus bacteria on the previous slide. They also have a different color on the light microscopy. So again, this is a Gram stain, whereas the Staph aureus was purple. This is E. coli actually shown here. So it has a pink color instead of being that deep purple, so it's Gram negative. This is a picture of Pseudomonas aeruginosa, which is another gram-negative bacteria. Again, you see it's kind of this rod-like appearance, similar to our Enterobacteriaceae, a little bit more elongated. All of these images are SEM, as you can see and recognize there. These are really notorious for being able to very quickly form a biofilm on a surface. So they're the most common offenders with catheter infections, urinary catheter infections especially, but they're also associated with what's known as hot tub rash. So if you Google that, you can see some really, frankly, pretty nasty pictures of skin rashes and infections that people can get if bodies of water like hot tubs or even swimming pools aren't properly maintained. Lastly, we have Candida as our representative fungi that can be infectious. So Candida is also known as yeast. It's really common in mucosal tissues, and it can thrive in low pH environments and in an inflammatory environment. So, of course, with that in mind, if we had Candida on the surface of an implant that's going into a tissue, during that inflammatory reaction, after the injury and the surgery, it could thrive here and survive, and that would be very problematic. So when we think about our implant-associated infections, we can classify them a few different ways. So really, this can be done based on the time of onset and then the location in the body. So we can have superficial immediate infections. So in this case, it's growth of microorganisms on the skin that is in association with an implant or soon after a procedure. So an example of this would be growth under a burn dressing. It could also be on anything percutaneous, like a percutaneous device, such as a hemodialysis catheter, for example, or if you had percutaneous pins in place for fracture fixation, for example. If you have an external fixator, if you've ever seen one of those. We can also have deep immediate infections. So this is infection at the implant site soon after surgery. But obviously this is deep inside of the patient. So this might be if you had like a knee implant and a patient develops an infection shortly after implantation that's associated with that device. So these are caused typically by skin-dwelling bacteria. So these are normally innocuous, but if they get transplanted to that implantation site during surgery, then they can begin to grow on that implant and cause problems. The third way we can classify it is late infection. And this is really probably the most problematic of the three. So with superficial immediate infections and deep immediate infections, as long as your device is sterilized and you sterilize the surgical site before you start making incisions, you should be able to prevent these. But the late infections, these can occur at an implantation site months to even years after a surgery is done. And the mechanism on this is a little bit unclear, so it's still an active area of investigation. But it occurs because it's the seeding of bloodborne pathogens that originate at another site. But then if they're able to make it to the site of an implant, and then because of an affinity for that implant surface, they can start to grow and thrive there at that location in the body. So this has been observed with orthopedic implants like knees and hips. Now let's talk about the steps to infection. So there are 4 steps here. So there's attachment, which is a reversible step. It's based on non-specific interactions between a pathogen and the surface. Then we have adhesion, which is irreversible, and that's a combination of non-specific and specific receptor-ligand interactions. So these two things are kind of similar to protein adsorption and then cell attachment. Once we get adhesion to a biomaterial surface, the next step is aggregation and biofilm formation. So the bacteria will divide and form colonies, and this can happen very quickly because bacteria divide very quickly. And then once they start to form these colonies, they exude an extracellular polysaccharide slime known as a biofilm. So that's one of our important concepts for this lecture. And then that biofilm actually protects the bacteria from phagocytosis. So it protects them from our immune system. And that's one way in which they're able to thrive. The 4th and final step is dispersion. So this can occur particularly if you have a device that is in the bloodstream. So this is probably not going to happen if you have a device that's in a tissue. But if you have a device that's in the bloodstream, so like a heart valve or a hemodialysis catheter, if you have the biofilm formation and then the shear forces can break off a portion of that bacterial colony and carry it to other areas within the body, so then you can get infection at a distant site from the implant. This figure illustrates these steps visually for you. So here at the top, we have step 1, our attachment. Step 2, you have adhesion. And you see down here, these are mediated by these reversible non-covalent interactions, so van der Waals forces and hydrophobic interactions. So in this figure, so B, that's a bacteria, and then we have our surface substratum down here on the bottom. That's our biomaterial. So bacteria gets close, we have the attachment and adhesion. So first non-specific interactions, and then adhesion, we get specific receptor-ligand interactions that facilitate that attachment. Adhesion occurs because bacteria, much like mammalian cells, they have receptors on their surface, and they have receptors for a variety of ECM components. And these are kind of analogous to our integrins on our cells, but they're called IMSCRAMs for short. So these are microbial surface components recognizing adhesive matrix molecules. So you can remember that IMSCRAM abbreviation and that they are like integrins. And so as an example, Staphylococcus aureus uses IMSCRAMs to attach to fibronectin, vitronectin, and von Willebrand factor. After adhesion, step 3 is our aggregation. So the bacteria start to divide. So you see now we have more of these circles with a B in them. Some of them don't have a B, but those are all the same. So they're dividing. They also form this polysaccharide slime. So this is our biofilm. It's being represented there. So those are polysaccharide chains. And then when we get shear forces here, the fourth and final step of dispersion can occur, and that will carry them away in the bloodstream. Now I want to talk a little bit more about biofilm formation, because this is a really important concept. So the biofilm, as I said earlier, so it's a polysaccharide slime, and what's very important about it is that it reduces the body's ability to kill adherent bacteria for several reasons. So one is that it's just a physical barrier to phagocytic cells. So this is going to inhibit T cell and B cell activation, inhibit antibody production, and inhibit bacterial opsonization and phagocytosis by our innate immune cells. So that's, of course, going to allow them to evade our immune system and thrive on the implant surface and within this biofilm. The second reason it's important and problematic is that it can impart antibiotic resistance. And the reason for this is kind of analogous to the problems that we talked about with drug delivery and fibrous encapsulation. So when you have this encapsulation around an area, it's a diffusion barrier. So in this case, the biofilm acts as a diffusion barrier to antibiotics, and so the antibiotics are not able to get in and kill all of the bacteria. Now, as I mentioned, this can happen very, very quickly because of the rapid growth rate of bacteria. So this slide I'm showing biofilm formation. It's from a research paper, and you can see here this is Staphylococcus epidermidis, so one of our common offenders, gram-positive bacteria that is found on our skin. And you can see up here, so these are the bacteria right here. They have the similar structure to the other picture that I showed before, so kind of these clusters of spheres, clusters of grapes, although these look a little less grape-like. These are, again, scanning electron micrographs. See, at 2 hours, we have that adhesion, right, some attachment, and then within 4 hours, they're starting to proliferate. So if we go back a couple slides here, so 2 hours, we're at adhesion, attachment and adhesion, and then here at step 3, so we're getting aggregation after just 4 hours, and then after 8 hours, you actually can't even see any of the underlying biomaterial anymore. So they're starting to grow that much, and then at 24 hours, we have a robust biofilm. So that's kind of what all that is. You don't even see all of the bacteria there because they're embedded in that biofilm matrix. And again, this is completely covering the surface of the biomaterial. So in this study, this was, colonization and biofilm formation of N-butyl 2-cyanoacrylate. So this is kind of like a super glue, but it's used as a tissue adhesive. So an alternative to sutures if you don't need as much strength. So sometimes these tissue adhesives can be used to close an incisional wound. But you can see here, if there's any Staphylococcus epidermidis around with this particular tissue adhesive, it can form a biofilm very quickly. So as we think about this process now of the steps that are involved in infection and getting to biofilm formation, you can think of it kind of as a race to the surface too. So there are soluble proteins. Protein adsorption still matters, just like it did in our previous course material when we talked about protein adsorption, how that drives the response of our cells. Well, the bacteria, they can also respond to proteins that are adsorbed, so that can dictate what bacteria will attach. And then, so the proteins, they can get there first, of course, because they're small. But then our surrounding cells getting on the surface, and then potential pathogens. So they're all kind of competing against each other. And if the pathogens win, then we lose and we have a device-associated infection. So with this in mind, we have to think about what proteins initially adsorb. That's going to drive the response. And then how these proteins interact with cells and bacteria. So, and again, that's important because of these imsgrams that bacteria can use. So with that, we will stop here on device-associated infections, and in the next video, we'll be going over methods to prevent device-associated infections.